Hello, anatomy and physiology students. This is Miss Carlson again. We are going to move on to Chapter 3, Cell Structure and Function, which is basically a review of about 3 different units from bio. So we're squeezing that all into one chapter. It's really important that if there's something that you're struggling to remember, post a question to the discussion on Schoology. Have a notebook, sheet of paper, or a template ready to take good Cornell notes, and we're going to go ahead and get started. 3.1 basically emphasizes that the study of cells is essential to understanding the physiology of our body. So it's basically that base, that foundation. If we can understand how all these different kinds of cells work, we can understand how the tissues, organs, systems, and then the entire body works. And you can see that there are so many different types of cells, and these are not even, this is just a very small portion of them. They're all different sizes, they're all different shapes, they have all different structures, and that helps provide the function that they need to sustain life. The cell theory states the following. Cells are the building blocks of all plants and animals because they are the smallest functioning unit of life. So they basically have all the characteristics of living things in one cell. They are produced by the division of pre-existing cells, and then kind of a tag on from what you might have seen in your biology textbook. Each cell maintains homeostasis or is able to regulate its internal functions. This theory basically tells you what cells are capable of doing and, again, what they do for us. So this is a typical cell diagram. It's not as detailed as some you've seen from your hat pack and in your textbook, but it's just a general cell diagram, kind of emphasizing what all cells generally have, and that they're surrounded by extracellular fluid, so they're usually suspended in some kind of fluid externally or on the outside of their boundary or cell membrane. So a cell membrane is also known as a plasma membrane, and that's just the outer boundary of the cell that keeps in all of the contents and separates the cell from the outside and from the cytoplasm. So the contents are suspended in the cytoplasm, which is just this fluid-like center. Again, just as the cells are suspended in an extracellular fluid, the organelles within the cell are suspended in a fluid known as the cytoplasm. It can also be called the cytosol. Now, the way we typically look at cells is with the use of an electron microscope if you are really fancy, or for us, we usually use a light microscope and can see most cells. 3.2 emphasizes the structure of the plasma membrane, which is very important. The functions of the cell membrane help us to maintain homeostasis, or I should say helps our cells maintain homeostasis. So it's a physical isolation, exchanges materials from in and out of the cell. It helps the cell have sensitivity to sense different stimuli, and then also provides structural support. There are 3 main components to the plasma membrane, and that's the phospholipid bilayer, which is made up of primarily lipids. You have carbohydrate chains. They're kind of like signals, signalers or receptors. And then you also have your proteins that are embedded within the membrane that usually allow certain molecules or substances to leave or come into the cell. 3.3 talks about how certain molecules and substances are able to cross the cell membrane, and this tells us, or basically reminds us, that our cell membrane is selectively permeable. Selectively meaning that it will allow some things to go in and out, and it won't allow some things to go in and out. This has also been termed semi-permeable. They mean the same thing. So a couple of ways that things can diffuse through the membrane are through diffusion. It's from a high to low concentration gradient, and basically fluids or substances, solutes, they'll continue to move until that gradient is eliminated. Osmosis is the same thing essentially, but we're specifically talking about the movement of the water, and that's due to some sort of osmotic pressure. And then there's also filtration, which can separate solutes from a liquid. We don't really talk about that too much. We'll really emphasize the understanding of diffusion and osmosis. So So in the picture over here, you can see that substances like water, small water-soluble molecules, can diffuse through the membrane through those channel proteins. Some large ones cannot diffuse through lipids and cannot cross the plasma membrane unless they're transported by some kind of carrier mechanism because they're too big. Some molecules are lipid soluble and can diffuse straight through. So again, our membrane is selectively permeable. Now I want to take a minute and just focus on osmosis for a second. This is usually an area of struggle for students in understanding the difference between a cell that is suspended in an isotonic solution. And notice that the arrows are going back and forth, meaning that the solute water molecules are moving freely. And we really just focus on the water when we're talking about osmosis, and that might help you understand this better. In a hypotonic solution, that means there's a less concentration of water on the outside compared to the inside. Notice that the solute here is making the water concentration less on the inside of the cell and more on the outside because there is no solute, so the water is going to move in. So I like to think of hypo, think of like an O, like looking like a mouth and taking things in and swelling up. And then hypertonic is the exact opposite. The concentration of water inside the cell is greater than the outside the cell because of some solute concentration. So in an effort to have that high to low flow, the water will leave, as indicated by the arrows, until the gradient is even out and the cell will shrink. In this case, you have a crenated red blood cell, which basically renders the cell useless for the body. Okay, so there are a few other types of transport mechanisms to get molecules, substances past the membrane, and so they're carrier-mediated or vesicular transport mechanisms. One of those is facilitated diffusion, which allows things to go through with a carrier protein, as shown in this picture here. Glucose is a very common molecule that tries to pass through that's too large, so this carrier protein will recognize that glucose molecule and open up and allow it to go through the membrane, access that carrier protein. And then another type of transport would be active transport. This is the one that requires energy. So we're going to consume ATP due to a low to high gradient. So you can kind of think of it as going up a hill. So energy or your pumps are needed to push whatever it is that's trying to get through or go past that low to high gradient. Finally, we have vesicular transport, and that's when materials move in or out through membranous sacs. And there are two types. There's endocytosis and exocytosis. Endo meaning things are getting taken into the cell, exo meaning items are going out. There are two types of endocytosis that we focus on. One is called phagocytosis, and that is cell eating, and penocytosis is cell drinking. It looks very similar, but there are some fine differences, and they are pictured in your textbook, and it was one of the questions in your hat pack, so make sure you look at that diagram closely. Okay, 3.5 gets into all the organelles, like the main ones, and there are specific functions. Now you'll find when we talk about each unit and we talk about the cells that make up the tissues and organs for that unit, that they don't all have all these organelles because they're specialized for that specific organ. So we just have a broad understanding of each of these organelles and know their functions. Some of the main ones pictured here are the nucleus. Remember, that's the control center of the cell. There's the endoplasmic reticulum. This is the rough because it has the ribosome studded on it. They are a transport highway for those ribosomes. And those ribosomes, remember, create proteins or synthesize proteins. You have these kind of circular objects. Some of those are lysosomes. Some of them could be labeled vacuoles. And some of them can also be vesicles, like that vesicular transport we were just talking about. It could be something leaving or coming into the cell. So those look very similar. And then your Golgi complex right here that modifies, sorts, and packages proteins for transport. You have, of course, your mitochondria that kind of look like hot dogs. I think the matrix and that inside portion kind of makes it look like a drizzle of ketchup. You know, that's why I think of it like a hot dog. Use whatever you can to help you remember them, but that is our energy source for the cell. And so those are just a few of them. Please carefully look over them. We'll do some practice with remembering the functions of these organelles, but I'm not going to talk about them each individually here today. Now, 3.6 focuses on the nucleus and how it contains DNA and enzymes to help the cell do a variety of functions. So the nucleus itself is surrounded by a nuclear membrane. There's a nucleolus in the center that also helps make those ribosomes. And then you also have some nucleoplasm, which is just like cytoplasm for the entire cell, but now we have that fluid inside the nucleus that's suspending the nucleolus and other structures. So it is the control center, like I mentioned a second ago, and the nuclear envelope helps communicate with the cytosol, or again, that also means cytoplasm, through pores. So things can leave the nucleus via the pores. Some other main functions of the nucleus is that it directs synthesis of proteins from DNA blueprints. So it takes the DNA within the nucleus and helps those blueprints get read so that proteins can be made. When those proteins are made, we're using a triplet code. You might have heard of them referred to as a codon, and that overall is called the genetic code to identify individual amino acids. And we're going to talk about that more in a second. But each gene's triplets on a chromosome will specify for a particular protein. Let's talk about this protein synthesis a little more. There were two processes that occurred to help create proteins. One was transcription. This is where the messenger RNA actually comes into the nucleus, matches up with an unzipped side of the DNA, and matches up those nitrogenous bases. So A would always go with T, but if A was matching up with the base of RNA, it would be a U, because RNA doesn't have thymine, it has uracil. And then C would always match up with G. So once it matched up with all those bases from that DNA blueprint, you would have this mRNA strand that is going to be read outside of the nucleus in a ribosome by each codon, or triplet is what I just mentioned to you. So once that mRNA leaves the nucleus, and you can see it pictured here, it's going to find a ribosome, and then tRNA is going to perform the translation function, and tRNA looks like this. It brings the anticodons to match up with the appropriate bases of mRNA, and what it ends up tagging along is the amino acid that's going to be part of building that protein. And each one of these amino acids will be attached by a peptide bond, and once you hit that stop codon, then it knows it's completed the protein, and it will stop and then make a new protein or start the whole process over again. So transcription and translation, we will really practice this in class and review it again, but that is the basic general understanding of how the two work. Transcription is reading the DNA, translating is writing and forming those amino acid chains. And this is that genetic code I was talking about. So if I had this DNA triplet or codon, it would match up with this mRNA codon. Remember I said RNA does not have thymine, it has uracil, so U would match up with the A. And then the anticodon for tRNA would again be the 3 A's. And you would read this mRNA codon though to translate phenylalanine, and that would be the first amino acid in this code. So again, if this sounds extremely unfamiliar to you, it's okay. We're going to practice this again in class, but this is just an example of the triplet code or the genetic code. Okay, last couple sections here, the stages of a cell's life cycle. This is a review of basically cell division and the entire cell cycle, where the main phases of the cell cycle are interphase, which is the longest phase and the phase that somatic cells are in the majority of the time. You have mitosis, which is the actual division part. And then you have cytokinesis. It's that final step after mitosis has occurred where the two cells separate completely. Okay, so cell division is a reproduction of cells, and there's also this genetically controlled cell death that can happen if there is a problem with the cell, and that's called apoptosis. So before division can happen, though, somatic cells must complete S phase, which, if you remember, if I go back to that picture we just looked at, S phase is DNA replication. So, DNA has to unzip and replicate so that you have double the amount of DNA, so that when those two cells separate, you have those two daughter cells, they come out with the same amount of DNA. So mitosis then would occur after that DNA replication, and so we have interphase, which is not a part of mitosis, it's a part of the cell cycle. But once mitosis begins, you have prophase, and it usually breaks them down into early and late. Early, remember, the nuclear membrane starts to disintegrate. You have the centrioles and spindle fibers that show up, and the chromosomes are going to condense, and you can actually see them in the copied pair. Then you have metaphase, where all the chromosomes line up in the middle and get attached to the spindle fibers. Anaphase, those chromatids are going to pull apart. Telophase, that cleavage furrow starts to pinch. And then finally, you have two daughter cells after cytokinesis, and then interphase would begin again. Okay, finally, 9 talks about tumors and cancer, and 310 talks about differentiation. So all I need you to generally know about tumors and cancers is that they are created because of out-of-control cell growth, and division will cause those tumors. And basically what's happening is, because the cell is dividing uncontrollably, it's creating this mass of cells, which we call a tumor. So they can be benign, which is non-cancerous, or malignant, which is cancerous and can invade other tissues. If it's determined that the tumor is malignant, then you are said to have cancer. Now, differentiation just talks about how cells are produced with specific limited capabilities. They don't start off this way. This happens as they grow and divide and start being specialized for creating special tissues and special organs. So cells that have differentiated and are organized collectively form our tissues, which is going to be the next chapter that we get into after this one. And I just wanted to show you a quick picture. Remember that cell differentiation, it starts with stem cells. So this is just a kind of a neat little breakdown showing you that we do start off with an embryonic stem cell that is totipotent. It can become anything, and then you go through these different levels of stem cells that are more specialized, and eventually we get to our individual types of cells that make up our tissues and our organs, like the lung, the pancreas, heart, etc. So at that point, these cells have differentiated completely and have special functions. The stem cells at these different points have not completely been identified with the function, so it's kind of interesting. Okay, that is it. Remember to go back and check out your need to knows, your prefixes and suffixes, general cell functions, and then the key terms in the Chapter 3 review, and I have provided those page numbers for you. Again, don't forget to post any questions that you have to that discussion on Schoology, and we'll see you again next time.